Good afternoon, everyone. My name is Mary Claire Fiddy. Over the past year, I've been working on researching and observing how the presence of contaminated microplastic beads affects grazing habits of copepods within mixed diets. There are many studies that prove that copepods ingest non-contaminated microplastics within a controlled environment, but none have shown how their habits are changed because of the presence of contaminated beads. Plastic pollution within the marine environment has been increasing over the decades and has led to concerns about the health of marine organisms and ecosystems. Microplastics, which are plastics less than 5 millimeters in diameter, enter the environment as primary plastics, which are produced to be small, and secondary microplastics, which form from the breakdown of larger plastic pieces. Plastics have unique properties, such as being hydrophobic and being lipophilic. These properties make them extremely durable as well as buoyant, but these properties also allow for the absorption of organic pollutants such as PCB and DDT. In earlier studies of copepods, microplastic beads were used to understand how copepods use their appendages to feed. This led to the realization that copepods ingest plastic, which led to more studies about how copepods eat plastic in mixed diets with their natural prey. They found that they will ingest microplastics, which can lead to a lack of mobility, as well as changes in life history, such as mortality and fecundity. Only one study has observed ingestion of microplastic pieces in an estuary, and this was done specifically in Tampa Bay. As the population around Tampa Bay increased in the 1950s, the use of pesticides such as DDT on people's lawns also increased. The bay became overrun with large amounts of pollution from the runoff, which led to large algal blooms disrupting the bay. Since the 1950s, the bay has improved greatly, and many of the pesticides used in the 50s are now banned by the EPA. But due to the long half-lives of the chemicals, the pollution still remains in the bay. Along with the pesticide pollution, it was observed in 2019 that there was microplastic pollution in the water column. With the amount of plastics in the bay, as well as chemicals, the microplastics could be absorbing the pollutants that remain in the microlayer. Laboratory and field studies show that copepods ingest microplastics, which has led to speculation of bioaccumulation of particles in individual organisms, which could lead to biomagnification through the food chain. The primary concern is that plastic pieces that store pollutants could be absorbed by the organism's body and move up the food chain, eventually to our plate. Miller 2020 reviewed all present research on bioaccumulation and biomagnification of microplastics and found that at this time there is evidence of bioaccumulation, specifically in lower trophic levels. However, there is no evidence of biomagnification. This, they believe, is because large animals have the ability to pass microplastics through their digestive tract. However, there are little studies on this topic, and that conclusion is early. Based on previous ingestion studies that show how copepods selectively feed on particles that are of high nutritional quality than low, I hypothesize that Acacia tonsa, the dominant copepod species in Tampa Bay, will exhibit prey switching behavior in treatments depending on the proportion of phytoplankton and non-contaminated beads. Next, I hypothesize that Acacia tonsa will select DDT contaminated microbeads no matter the ratio of microbeads to phytoplankton in the treatment diet. Previous studies indicate that copepods avoid contaminated or toxic phytoplankton based on chemical signals. I predict the copepods will act the same way to contaminated beads. All 5 of our experiments started in Tampa Bay, collecting samples from Boca Ciega Bay and in front of Indian Key, which can be seen on the map in the middle of the slide. Copepods were collected with a 200-micron mesh net at both stations. The plankton tow process can be seen on the images on the left and on the right. Before each grazing experiment, Acacia tonsa was starved for 24 hours, and batch cultures of a centric diatom and microbeads were prepared. After 24 hours of starvation, treatment bottles were made. This graphic shows how we set up our individual treatment bottles. Large batches of 100% phytoplankton and 100% microbeads were used to make mixed diets of different treatments, which can be seen in the center of the graphic. There are 5 replicates per treatment with a total of 5 treatments. 10 copepods were added to the replicate bottles. Once the bottles are ready, they were immediately added to the plankton grazing wheel, which can be seen on the right. The grazing reel is then placed in the incubator, constantly running for 24 hours. After the 24-hour grazing period, a flow cam was used to visually image similarly sized and individual phytoplankton and microbeads for quantification. Examples of what the grids from the flow cam look like are shown on the right of this slide. Oops, sorry. The top image shows the phytoplankton grid and the bottom shows the single microbead grid. This table shows a summary of the results gathered from 3 of the experiments that contain non-contaminated microbeads. Focusing on the red box, the grazing rate in our study is consistent with other studies which show higher ingestion and lower concentration of phytoplankton. The clearance rate ingestion rate of particles per day is much lower compared to other studies. I believe this to be because the other studies use larger copepods, different phytoplankton, longer grazing times, and some do not contain mixed diets with microplastics. An important takeaway from this table is that in all categories, including grazing rate per day on phytoplankton, clearance rate of phytoplankton, and ingestion rate of phytoplankton was all higher than that with microbeads. This shows that copepods are selectively feeding phytoplankton cells that have higher nutritional value than beads. Previous studies came to the same conclusion. When looking at the ingestion rate ratio of phytoplankton and microbeads, the dark gray linear line indicates a 1-to-1 ratio. The points above the line, the areas outlined in green, indicate that more phytoplankton are being ingested. And points below the line, the areas marked off in blue, indicate more microbeads are being ingested. Each point represents the mean from each experiment. So for each treatment, there are 5 points. The graph shows that all experiments with non-contaminated beads, more phytoplankton were ingested by copepods than microbeads. On the other hand, there were 4 instances where contaminated beads were ingested over the available phytoplankton. Our results show when contaminated beads are offered with phytoplankton, the results are mixed. That ingestion of contaminated beads is higher than phytoplankton, but not in all. I also observed organic material around the contaminated beads that could make the beads more desirable to copepods. Similar conclusions have been made when looking at plastic ingestion in seabirds. There are several important takeaways. The first is that copepods ingest both contaminated and non-contaminated beads. This means they do eat plastic. Also, that contaminated beads are ingested at a higher rate than phytoplankton. And in all cases, phytoplankton were ingested more than non-contaminated beads. The implications of this study are that similar situations could be occurring in Tampa Bay. It is known that there are dangerous pollutants found in the bay and microbeads present. Also, that copepods within the bay are ingesting microplastic. This means that copepods within the bay could be ingesting contaminated plastic at a higher rate than phytoplankton present. It is not known why they ingest contaminated beads and how the ingestion affects their physiology, as well as if copepods absorb the contaminants in their tissue. These are all areas that need further study. I would like to acknowledge Yakugawa Fluid Imaging Technology Incorporated for providing the FlowCam, and Savannah Judge for technical assistance while using the FlowCam. I would also like to acknowledge Eckerd College for providing funding and research and lab materials. Thank you all for your attention and time. I would like to open the floor up for questions from the audience.